Last talk today. One group of you will attend our imaging center later this afternoon, and before you will touch the animals, I will just briefly let you know what are the rules, procedures, and so on. So. Oh. Okay, so why we are working with small laboratory animals? You know that modern laboratory research, biomedical research, use many omics, genomics, proteomics, metabolomics, and it brings a huge information about functioning of the cells and organisms, as well as of causes of diseases and possible approaches to cure them. Also very important is translational research, which brings this knowledge from the laboratories to clinics, from bench to laboratory bench to patient's bedside. And what is very important that nowadays we have animal models which mimic almost all important diseases, human diseases. There is a lot of mouse models of human diseases, models for obesity, for diabetes, for neurodegenerative diseases, autoimmunity, a lot of models of leukemias and tumors, and we also have humanized mice. We grow human cells, immunodeficient mice, and for example, such mice can produce human blood cells. And the top is patient-derived xenografts. If you transplant cells from the particular patient into immunodeficient mice, and you can use different approaches to treat the particular disease. We have many mice strains with reported genes. I was talking in the morning that we use these mice mostly for regenerative medicine. When we transplant stem cells, all the progeny of transplanted cells will bear also the reported gene, and we can show how the regeneration works. Green is not very good for in vivo imaging, so we prefer red mice, and if you don't like these colors, there is plenty of different animal colors. There are even red dogs, fish. If we look at Nobel Prizes for physiology and medicine category, since 1901 there was awarding 115 awards. Not all years during the war the awards were not awarded, so it's less than number of years. And out of these 115 awards, recipients 103 used animal models in their research, and virtually any Nobel Prize for physiology or medicine during last 30 years use animal models. If you want to test preclinically something on laboratory animals, how you choose your model? It depends on your laboratory project and on the experience of the laboratory, history of the laboratory. Of course, very important is are possibilities of animal facility. Which animals are they able to breed or maintain? You search for your topic in the literature, and maybe you might find interesting model that you did not try yet. Of course, very important is size of the animal. Most experiments are done on mice. Mice are small, so if we want to image or to do some surgery, or it's easier on bigger animals. So we use rats, and cardiologists first used dogs. The rabbits are used as well. And of course, don't forget the physiology, because it is not always transferable. For example, translatable. For example, mice have different immunity compared to man. Human are based mostly on innate immunity. So they have a lot of granulocytes, macrophages are present, and so on, and they have less lymphocytes that have mice. You know that mice live in a very dirty environment, and they fight together. There is a lot of blood, and they are wounded, and in fact there is no lethal infection among them. So they heal everything much better than we do. So it is not necessary, really necessary, to work strictly sterile if you do some mouse surgery. Of course, we do it because we want to be very careful. But if you will not sterilize or your sizers and so on, so it will the mouse will not die. Federal Drug Administration in the United States set up the 2-animal rule. That it's not valid anymore, but for many decades was mandatory that all vaccines had to be tested for efficacy and safety in 2 animal species, usually on a rodent model, which is cheaper and easier to work with it, and also before introduction to humans, they prefer to test also on a non-humanid primate. Macaques or something like this. We need animals for research and education to understand how disease or injuries function, affect living organisms, and to develop the cure for the diseases. Animals provide such an index of safety, because already in 1949 was mandated that animal studies should precede and support human studies. And in Helsinki Declaration in 1964 was stated that medical research on humans must be supported by preceding animal research. You can see that mice and rats are about 95% of all laboratory animals use small number of dogs and cats, and even less non-human primates. Other species are other rodents, rabbits, fish, sometimes amphibians or reptile, and also livestock, pigs, cattle, sheep, goats. It's because of their size. And if you look for numbers, how many animals are killed by different circumstances. So research animals, except mice, rats, and birds, it's 1 million per whole year. research including mice, rats and birds, 26 million per year. But hunting kills 150 million animals, and automobiles kill 1 million of animals every day. Ducks for food, the number of ducks killed for our food is comparable to all number of research animals that we use. Cattle, sheep, cows for food, even more. Pigs, 4 times more. And the biggest amount of animals are killed for food are chicken. Work with experimental animals was first reported in ancient Greek and in small extent in ancient Rome. In that time, Galenus was a physician who described different procedures, how to handle animals, how to make some surgeries or everything around, and these books were then canonical, undisputable until Renaissance. Nobody used experimental animals after ancient Greeks and Romans, but in 16th, 17th century, when Renaissance came, there appeared some scientists who again set up the modern comparative anatomy, and they described this. Harvey, described the bloodstream and function of heart and veins in the body. Vesalius. Even the start of bigger use of laboratory animals was in 20th century, and Barnard first before heart transplantation, he transplanted heart to the dog, and end of the last century we expected that very much such experiment could be replaced in silico by computing, by in vitro techniques. But it appeared that it was not true, because at that time we discovered the genome. Mouse genome was completely described just before human genome. There are knockout mice produced which have knockouted every gene for every protein in the body, and they phenotype these mice and look how function this protein, and for different combinations, and of course. Genetic manipulation prepares for every major disease the animal model. So now the use of experimental animals is again increasing. European legislation was set up in 2010 by directive number 63, and it set up legal standards for protection of laboratory animals across all European Union. It covers all aspects of animal breeding, animal use, from housing to food and care and approval of research projects. This directive was applied to all European countries. In some countries, it's easier to work with laboratory animals. In some, it's very complicated. For example, Germany. It's very easy to apply experimental treatment to humans. the physician just need informed consent. But the same is not possible to do with mice. So we have a lot of collaboration with German scientists because it's easier to do animal experiments here. The key pillars of this directive include classification of procedure severity. If it's mild, non-recovery mild, moderate, or severe. If you do the experiment on animals, finally you had to kill the animal. It's a rule. Mandatory is that every scientist should apply for, should write his animal project, and it must be evaluated and authorized. There are strict requirements for staff training and competence, and also every institution has to establish a local animal welfare committee body. So each European country transposed this directive into national law, and for you it's mandatory to know how it works in your country, and you must do everything what is required by this directive. Before you start to work with animals, you must prepare a detailed project proposal. You must submit it to the animal welfare body in the institute, and after they will agree with it, it is sent to the ministry, and they have reviewers which can ask you why do you do this and that, and they can even push on you to change the severity of experiment or number of used animals. It usually takes very long. In our country, it's minimum 3 months to obtain the permit to work on the particular experimental setup, and in some countries it's 1 year. We have in our core facility, we have a general approval for preliminary research, because before the scientists apply for a grant, they need some preliminary results. So we have approval to make on small groups of mice very broad spectrum of experiments. So in this case, we can react very fast. But if they get a grant, they have to apply for the full approval. There is a 3 R's principle: replacement, reduction, and refinement. Replacement: if it is possible, don't use animals. Reduction: use minimum number of animals to validate your results. It's not easy. If you speak to statisticians, they usually prefer to have hundreds of results to make valid statistics, but we use groups of 5, 6 mice. Refinement: minimizing any potential pain or distress and enhance animal welfare. The proposal must include non-technical summary for the public, that everybody will understand why is it done. Clear scientific objectives and justification. A thorough explanation of how the 3 R's are applied in the project, and very detailed explanation, a description of all procedures, number of mice, number of groups, what will you do with them. And finally, you must define endpoints for the experiments. When you will kill the animal, when you will finish the experiment. It's not allowed to inject cancer cells and let the mice to die for cancer. You must kill it when the tumor exceeds 10% of the body weight, mostly. Animal welfare body is a local committee within each institute, and it oversees animal welfare and ethical compliance. It must include animal welfare officer, scientist, and veterinarian. All personnel who are working with animals should pass the 1 week course with examination at the end, and they must be properly educated, trained, and supervised for all the experiments. And if you pass this course, you can write the proposal for animal experiment, and you can do experiments also on animals. We mostly use laboratory mice. It's a small animal. It's relatively cheap. It's relatively because one mouse can cost even several $100s, euro for if it is special GMO strain. It has a rapid breeding, and we understand the genetics. We know all the genome, and so it's the most common mammal in biomedical research. For some experiments, we use laboratory rats, which are larger and stronger and much more clever than mice. They communicate together with ultrasound. You don't hear it, but they can, and then it's very important to allow rats to see one another and to have a way to communicate. They are widely used in many types of biomedical research and also in some surgery. And there are different strains and stocks of mice and rats. Inbred strains are genetical twins. One egg twins. They are genetically completely the same. So you can expect that the variability of results of the treatment or biodistribution or anything else will be quite homogeneous within inbred strains. For some experiments, you need outbred strains or stocks. They are genetically variable, and it's much more close to human diversity. For example, Vista rats. The inbred strains, which are mouse inbred strains, which are most used in the work, are this C57BL6 strain. And we have also genetically altered or genetically modified animals or organisms. We have models where specific genes which are added or removed or modified, and you know that it's now very easy with CRISPR-Cas9 technique. If you work with animals, you handle them, and handling means that handling of animals influences behavior of the animals. There are different animal handling techniques for mouse: blood collection from tailway, orbital sinus, cardiac punction, from saphenous vein, IP, intraperitoneal injections, subcutaneous injection, oral feeding, and also you must be trained in sexing mice. If they are very young, there is no difference between male and female, but at the age around 5-6 weeks, you can already distinguish it. But you must be quite experienced in this. It's not very easy. If you want to buy animals or obtain animals, it must be from licensed breeders, licensed breeding facilities. These suppliers have to follow very strict rules about the specific health status of the animals. Most commonly, we use SPF animals, specific pathogen-free. There is a defined set of pathogens which are not allowed to be in such animals. There are also gnotobiotic animals which don't have microflora at all. It's not very common situation, but for some types of experiments they are used, but the use is very minor and very special, and not many laboratories can do it. For animal transportation, you also will need to have a permission, and for example, mice have been sent in a filtered, climate-controlled containers, boxes with food and source of hydration. You will not put them water, but there must be some gel or fluid, and they can be transported just with approved couriers. You cannot use the FedEx or it must be special approved courier, but be sure if you will send GMO animals, there is not much such approved couriers, and many of them they can transport animals are not approved for GMO, so be careful on this, because they can be very big fines if you don't follow the rule. The cage is microenvironment for mice. It's their home. Must be made of transparent material to observe animals if they are okay, and they have to have a secure lid that the mice or rats cannot escape. And there are rules how the height of the lid for rats and for mice is minimum height that you must use. There are in the declaration there are size square centimeters per animal. So usually in such a cage you have maximum five mice. And the animal facility, vivarium, it's a macro environment. The animal rooms are to be strictly controlled for temperature, which must be between 20 to 24 degrees Celsius, humidity. Typically, you have set up a light cycle, 12 hours dark, 12 hours light. The light should not be too strong, because they prefer, it can be stressful for them. And ventilation, they must have 15 to 20 air changes of the atmosphere in the room per hour. The gold standard now for maintaining and breeding of laboratory mice and rats are individually ventilated cages. You have a special filter unit and the rack and tube which has air input and output. and you put there the box with animals, and there goes filtrated air and in and also out, and it assures that you cannot cross-contaminate animals. If some animal will have some infection, it is not easy to contaminate animal in another cage. And if you change bedding or food, or you manipulate with such mice and rats, it's done usually in the laminar hoods. Bedding and nesting material. It's very essential. Beddings are wood shavings, corn cob, to absorb the waste and allows for digging. Nesting material, paid with strips, cotton squares, is important for animals to make a nest, for warmth and for security. Also, the law set up the environment enrichment for animals. You should use such toys for them, just to give them possibility to do something, not just wait in the cage. Diet is very defined. It's low protein, high protein, low fat, high fat. They are usually made pellets, and then according to your experiment, you might have different nutrition requirements for your animals. Water bottles should never be dry, but be careful, because if you will put the water bottle too close to the bedding or a lot of bedding, there can capillarity take off the water from the bottle, and there will be a flooding in the box. So really it should be everyday controlled, that everything is in order. Husbandry routines, changing, cage changing, cage are changed in regular intervals, for example weekly, depends on the number of mice which are, or rats which are in the box. And it's done in a flow cabinet or biosafety stations. Different animal facilities have a different level of biosecurity. It's called animal biosafety levels, ABSL, to protect both animals and people. There are 4 levels of ABSL. ABSL 1 is for work with maybe potential pathogens, agents that are not known to cause disease in healthy adult humans. It's most labs, most animal facilities are in this regimen. You just wear laboratory coat and gloves. ABSL2 allows use agents that pose a moderate hazard. The free access to the lab is restricted. They must allow you, if you don't work there, they must allow you to go in. Work with infection materials is all done in class 2 laminar hood. ABSL3 is used for indigenous or exotic agents that may cause serious or potentially lethal disease, also via inhalation. It requires very specialized design and protective equipment, including respirators. And the top laboratory is ABSL4. It's for work with animals infected with dangerous or exotic agents that pose a high individual risk of life-threatening disease, aerosol transmission, or if you don't know what risk is from transmission of the related agents, and you have it's like a scaphandre with air supply from outside. So the rules to work there are very, very strict. Every animal colony should be monitored for health, biosecurity. Health monitoring. You must every 3 months from every room or cabinet or you must take one mouse. And then the mouse should be sent for dissection and a collection of blood for PCR and they check. This rule was set up by FELASA. It's a Federation of European Life Science Organizations. It's an organization that covers security, biosecurity in Europe, and there is a list of pathogens that must be tested every 3 months, or broader list every year. And we are not allowed to get animals from any other facility without such examination. Usually it's done by so-called sentinel animals because you don't want to take animals from your experiment. So you may put there one healthy mouse which is not treated, but it stays inside the cage with the animals, and if there is infection, it's infected. So it's also quite expensive to pay for all these tests. You must look for signs of healthy animal and signs of unhealthy animals in pain or distress. And you can healthy animals it looks like bright, alert, and responsive animal. Clear eyes, smooth and well-groomed coat. Normal posture and movement, unhealthy animal will have a piloerection, ruffled or unkept fur, a hunched posture. You can see the posture of healthy and ill animal. They may be in lethargy, isolated from the group, they lose weight, they can be dehydrated, and they have also some facial grimacing that you can learn. It's in human, and in mouse you can also observe similar grimaces. At the end of the experiments, you should euthanize the animals. There are predefined criteria in your project proposal. For example, certain tumor size, losing more than 20% of body weight, inability to eat or drink. And for euthanasis, it's used either inhaled agents. For rodents, it's mostly used 100% carbon dioxide. It's not allowed for primates to use the CO2. Because they have different biochemistry, and they are it activates anxiety, and also for us, if you have excess of carbon dioxide, you don't feel well. But for mice, it's used injectable agents, overdosing of anesthesia. Or physical methods mostly are used cervical dislocation. You use such a device or pencil, and you push the tail, and you dislocate the cervical vertebra. Handling and restraint. If you want to inject mice or collect blood, you need to capture it, usually for the tail base, and with second hand to the skin behind the neck, on the neck. Rats, you use such a technique that immobilizes them. You don't use, you can't transfer rats by taking them at the tip of the tail. Anesthesia. Anesthesia is mandatory for any procedure that can cause more than brief minor pain. It must provide unconsciousness and also analgesia, pain relief. And animals should be monitored throughout anesthesia. Very important is to heat them, because they are much more dependent. Rodents are much more dependent on production of heat by muscles. So if they are in unconscious, they don't produce enough heat, and they are hypothermic. Mostly we use inhalation anesthesia. Mostly we use isoflurane gas mixed with air or with oxygen, and we have such a vaporizer, specific vaporizer, and controlled induction chamber. When we put there is atmosphere of anesthetic gas, and we put there for a few minutes the mouse, and it sleeps. We also need to have a waste gas disposal. Either there are such filters that accumulate the excess of anesthetic gas, or in our labs we have exhaustion outside from the building. With isoflurane, we can anesthetize mice up to, let's say, mice or rats up to 2 hours, but depends also on the strain. Immunodeficient mice are more sensitive and might die. Sometimes we use injectable anesthetic agents, dissociative anesthetics. Typically, we use ketamine and xylazine, and it's very safe anesthesia injection by intraperitoneal injection, and anesthesia takes 1 hour, 1 to 2 hours maximum. But sometimes if you are doing some surgery or something like this, it's easier than when the mouse has a cone on a nose with anesthetic gas. What is also important to apply eye lubricant to prevent corneal drying if the anesthesia will take longer, and also heat source to prevent the hypothermia. Routes of injection administration. The common routes include subcutaneous injection, so you just inject mostly under the skin on the neck. Intraperitoneal injection, it's very rapidly absorbed via intestine. All the drugs or compounds that you inject this way, almost as fast as intravenous injection. And sometimes, if you want slow utilization of the drug, it's injected intramuscular. it's released for a longer time. Even there are such osmotic pumps. There are small capsules which can be implanted intraperitoneally, and they release the molecules of certain size for a prolonged period, for hours, even for days. So if you want to have a stable concentration of some chemical, it can be done also this way. What I want to point out that many chemists have no idea if their drugs or nanoparticles or anything are biocompatible. They must be, the drugs should be dissolved in water environment. We cannot inject, if it's is dissolved in ethanol or DMSO, we can use just very small amount of these organic solvents because they are toxic. You must you must first test all your solutions before injecting to mice on cells for biocompatibility, and then they should be non-toxic. You must adjust the pH. And also problem comes with viscosity. Sometimes they bring us the solutions which are enable to inject intravenously. It is not possible to pass it through thin needle. Blood collection. Often we need to collect blood, even repeatedly. The easiest way and fastest way is collection from retroorbital sinus, which is behind the eye, and you put such a microhematuric capillary tube. You rotate it and put it to the under the eye, and then you can collect the blood to Eppendorf tube, and it's very fast and very easy. You can anesthetize animal for a short time and collect as many blood as you need. Normally, if you are just screening something, single collection shouldn't exceed 10% of total volume, and total volume of blood in mouse is about 1.3 to 1.2 milliliters. So it's not much. Well, finally, we are imaging many mice and rats. Some laboratories are happy, more happy than we do. So it's nice to have access also to animals from zoo and so on. So thank you for your attention, and I hope that you will enjoy the afternoon lab tour and hands-on on MPI. And did you split into groups? Because all the people shouldn't. So you know who goes there. Okay, so if you have any questions regarding animal use, just feel free to ask. Okay, if not, so thank you for attention.